Last year, a team of biologists and computer scientists from Tufts University and the University of Vermont, UVM, created novel, tiny self-healing biological machines from frog cells called Xenobots that could move around, push a payload, and even exhibit collective behavior in the presence of a. Get ready for Xenobots 2.0. The same team has now created life forms that self-assemble a body from single cells, do not require muscle cells to move, and even demonstrate the capability of recordable memory. The new generation Xenobots also move faster, navigate different environments, and have longer lifespans than the first edition, and they still have the ability to work together in groups and heal themselves if damaged. The results of the new research were published today in Science Robotics. Compared to Xenobots 1.0, in which the millimeter-sized automatons were constructed in a top-down approach by manual placement of tissue and surgical shaping of frog skin and cardiac cells to produce motion, the next version of Xenobots takes a bottom-up approach. The biologists at Tufts took stem cells from embryos of the African frog Xenopus laevis, hence the name, Xenobots, and allowed them to self-assemble and grow into spheroids, where some of the cells after a few days differentiated to produce cilia, tiny hair-like projections that move back and forth. Instead of using manually sculpted cardiac cells whose natural rhythmic contractions allowed the original Xenobots to scuttle around, cilia give the new spheroidal bots legs to move them rapidly across a surface. In a frog, or human for that matter, cilia would normally be found on mucous surfaces, like in the lungs, to help push out pathogens and other foreign material. On the Xenobots, they are repurposed to provide rapid locomotion. We are witnessing the remarkable plasticity of cellular collectives, which build a rudimentary new body that is quite distinct from their default. In this case, a frog. Despite having a completely normal genome, said Michael Levin, distinguished professor of biology and director of the Allen. In a frog embryo, cells cooperate to create a tadpole. Here, removed from that context, we see that cells can repurpose their genetically encoded hardware, like cilia, for new functions such as locomotion. It is amazing that cells can spontaneously take on new roles and create new body plans and behaviors without long periods of evolutionary selection for those features. In a way, the Xenobots are constructed much like a traditional robot. Only we use cells and tissues rather than artificial components to build the shape and create predictable behavior, said senior scientist Doug Blackiston, who co-first authored the study with research technician Emma Lederer. On the biology end, this approach is helping us understand how cells communicate as they interact with one another during development, and how we might better control those interactions. While the Tufts scientists created the physical organisms, scientists at UVM were busy running computer simul. Using the Deep Green supercomputer cluster at UVM's Vermont Advanced Computing Core, the team, led by computer scientists and robotics experts Josh Bingard and under hundreds of thousands of random environmental conditions using an evolutionary algorithm. These simulations were used to identify Xenobots most able to work together in swarms to gather large piles of debris in a field of particles. We know the task, but it's not at all obvious, for people, what a successful design should look like. That's where the supercomputer comes in and searches over the space of all possible Xenobot swarms to find the swarm that does the job best, says Bingard. We want Xenobots to do useful work. Right now we're giving them simple tasks, but ultimately we're aiming for a new kind of living tool that could, for example, clean up microplastics in the ocean or contaminants in soil. It turns out, the new Xenobots are much faster and better at tasks such as garbage collection than last year's Mo. They can also cover large flat surfaces, or travel through narrow capillaries. These studies also suggest that the in silico simulations could in the future optimize additional features of biological bots for more complex behaviors. One important feature added in the Xenobot upgrade is the ability to record information. Now with memory, a central feature of robotics is the ability to record memory and use that information to modify the robot's actions and behavior. With that in mind, the Tufts scientists engineered the Xenobots with a read-write capability to record 1 bit of information, using a fluorescent reporter protein called EOSFP, which normally glows green. However, when exposed to light at 390 nanometers wavelength, the protein emits red light instead. The cells of the frog embryos were injected with messenger RNA coding for the EOSFP protein before stem cells were excised to create the Xenobots. The mature Xenobots now have a built-in fluorescent switch which can record exposure to blue light around 390 nanometers. The researchers tested the memory function by allowing 10 Xenobots to swim around a surface on which one spot is illuminated with a beam of 390 nanometers light. After 2 hours, they found that 3 bots emitted red light. The rest remained their original green, effectively recording the travel experience of the bots. This proof of principle of molecular memory could be extended in the future to detect and record not only light but also the presence of radioactive contamination, chemical pollutants, drugs, or a disease condition. Further engineering of the memory function could enable the recording of multiple stimuli, more bits of information, or allow the bots to release compounds or change behavior upon sensation of stimuli. When we bring in more capabilities to the bots, we can use the computer simulations to design them with more complex behaviors and the ability to carry out more elaborate tasks, said Bingard. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.